Visualization for the comparison of representatives. Attention please. If you have not seen the previous video tutorials, please watch them before you continue with this one. Hello and welcome to the video tutorial for JADOPPT for the visualization for the comparison of representatives. This video tutorial shows the different options that JADOPPT provides to analyze results, as well as the interaction between the molecular visualizer and the visualization. The visualization has two parts. The top one shows the general dendrogram, which represents the grouping of the representatives, and the bottom one shows the zoom group visualization. Most of the analysis of docking results consists of a great number of representatives. That's why this visualization has been included. Also, it is a detailed analysis. The top visualization shows on the left side the cluster per level that the bar cuts the dendrogram. These change according to the level you cut. As well, we can see some lines with different colors. These represent the reference structures which are known to have an affinity to the receptor. At the end of the dendrogram, it shows a block of different colors. They correspond with the color code on the left side. The red color shows the structure with low affinity, and the green color shows the ones with high affinity. The representatives which are in the same cluster with the references are expected to show a higher affinity than the ones that are further away. To analyze the clusters, we can click on the labels on the left side or on one of the branches. As we can see, they have changed to red, indicating that they are selected for the analysis. It is not until now that the visualizer shows us the cluster. As we advance, we will see the options of visualization of the molecular visualizer. At the bottom, we can see the zoom group visualization. As we have seen, it appears the selected branch. On the left side are the elements that form the cluster. They are labeled with the name of the original DLG file and the group number they represent in the DLG file. Note that the elements that are blue and start with R are the references. We can click on the labels and then observe them in the molecular visualizer. If we want to compare the grouped elements, we click on the circles that are next to the labels. Another interaction in the visualization zoom is the selection of sub-branches of the selected cluster. For example, as we select this branch, we can see that the visualization changes and that the sub-dendrogram is reduced. At the same time, we see that in the main dendrogram, one part, which had been red, changed to yellow, indicating that we are exploring other subgroups of the main cluster. This is linked to the molecular visualizer thanks to the interaction that exists among all visualizations. The buttons with arrows that point to the right and left are a browsing history of the subgroups. We can go backwards and forwards and see how the subdendrogram changes. The menu File serves to save the current visualization to continue working in it later. Another function of this visualization is to generate new references. For example, we select this group and now we select this element to generate a new reference. We name it as we like, and as we can see now, we have a new reference file. We can do the same but with the whole cluster. In the menu Clusters, you can find the option Compare All for a comparison of groups. Clicking, it selects all circles, and as you can see in the molecular visualizer, the groups changed color. To stop the comparison, just click on the option Clear Compare Selection. To determine where the elements of one particular DLG are, we apply the option Representatives from DLG of the menu elements. Clicking on that option, it will show us a window where all the processed DLG files are. As we click on one of them, we can see that on the right side it shows us the representatives of the groups of that DLG. Besides, we see in the dendrogram that some lines changed color. These are the elements of the DLG. By marking 1, the dendrogram changes color to highlight the elements. In the dendrogram, we can see the elements that represent the different groups that were formed in the first step. By unselecting the DLG, the dendrogram goes back to its original color. JADOPPT has an option to reduce the number of representatives without doing the first step. To do this, we click on the button Recluster Representatives. In this window, we have various options. First, we have two sources of representatives, the original representatives, or, if we already applied this option, the current representatives. Afterwards, we see four options that can be set to reduce the number of representatives for this recluster. For example, for the option RMSD, a distance of 3.0 is set, which enables the selection of those representatives that are at a distance of 3.0, reducing the quantity of representatives. At the same time, at the bottom, a before and after of the quantity of representatives is shown to enter the second step. The following option selects those representatives that are in a range of binding energies set by the slider. The third option selects the first 5 representatives with the highest binding energy. In this option, various DLG can remain left out, as well as in the previous option, and the same applies for the last option, where the representatives are selected by the number of clusters that are in the DLG. For this example, we select the RMSD with 8.0. Then we click on Accept, and we are asked to name the new visualization. We write RMSD, click on OK, and the window of the second step appears. We leave everything as it is and click on Accept, then we can see the window of the second step. As we finish, a message appears to tell us where the new result is saved. In all cases, they are in the menu of the window of the molecular visualizer, Visualizations, and in the submenu, Dendrograms. We click on OK, and now we can see the result where it was indicated. As we can see, this is the result of filtering the representatives. From here on, we can continue the analysis as we did it before. This is the end of our video tutorial, and we refer to the next video tutorial, Molecular Visualizer of JADOPPT. We hope that this has been helpful. See you soon!